So chapter 10, we're going to be talking about cell biology. So this slide just illustrates how small the cell really is. You don't have to memorize anything from this slide, but if you look in the middle between 1 micrometer and 100 micrometers, that is the size of the cells. So here again, your learning objectives. Make sure you can answer all of them completely. Try to go back and answer each one without looking if you can. So it's just a picture showing the prokaryote cell and the eukaryotic cell. He's not going to make you label anything, but again, it's just for illustration purposes. Notice that prokaryotic cells have no nucleus. So again, more pictures. Don't need to memorize anything from here. So you don't need to particularly memorize anything. Again, he won't make you label things, but this is an important picture. Make sure you know all the different compartments. We're going to be talking about each of them in this chapter. So just sort of know where they belong in the cell, what they're going to be doing. Just get a rough idea. So your very first learning objective is to describe the structure and composition of the plasma membrane. This is a very important slide. He's going to break down each of these chronologically. So we're going to start with the plasma membrane. So remember, your plasma membrane is what holds everything together. It allows things inside the cell and allows things out of the cell. So again, he doesn't show you pictures on his test and have you label them like in anatomy, but you should be able to visualize these when you study for the test. So here we have the plasma membrane. It's made of phospholipids. You should know the heads are hydrophilic, so that's on the outside. You should know that the tails are hydrophobic, which is in the inside. We have the glycocalyx on the outer surface. We have integral proteins, which is integrated right into the plasma membrane. We've got peripheral proteins that can be additions to the protein or the plasma membrane. We also see cholesterol in there. So you should know all these different properties. If I ask you, what are the properties of the plasma membrane? It has a lipid bilayer. It follows a fluid mosaic model. And then you should know what enhances fluidity. So that would be increase in temperature and increasing your cis unsaturated fatty acids. And then what can diminish fluidity? So cholesterol and long-chain saturated fatty acids. So this is still part of that first objective, knowing the composition of the plasma membrane. So remember, the plasma membrane is made of lipids, right? So particularly, they're called phospholipids. And you have an outer leaflet and an inner leaflet. You should know what type of phospholipid will I find in the outer leaflet. So phosphatidylcholine and sphingomyelin would be a phospholipid in the outer leaflet. If he asks what type of phospholipid will I find in the inner leaflet, then it would be phosphoserine and phosphatidylethanolamine. You're also going to find cholesterol. That's found in both leaflets, so don't be tricked. And then you're also going to find something called a glycolipid in the outer leaflet. So glyco means sugar, right? So it's just a lipid with a carbohydrate attached to it. If we were talking about glycoproteins, then it would just be protein with the sugar attached to it. So over here we have some examples of the glycolipids you're going to find in the outer leaflet. You should be able to tell me, okay, I'll find cerebroside, other neutral glycolipids like blood cell antigens A, B, and O, and then also gangliosides would all be found in the outer leaflet. The pictures on the bottom, he's not going to test you on, so you don't have to worry about that. So here we have a picture. For some reason, picture B didn't show up. But anyways, this is still going off of the previous slide. So remember, the phospholipids, they have a head group and they have a tail group. So the head group, if you look in picture A, is going to be your polar part, right? So that's going to be hydrophilic, and those are on the outside of the plasma membrane. On the inside of the plasma membrane, you have your hydrophobic tail. So if you look at these two in picture A, you have phosphatidylcholine and sphingomyelin. Remember, those are on the outer leaflet. And then in picture B, it's not shown, but it will be phosphoethanolamine, phosphoserine, and phosphoinositol. So just know your different head groups, but he's not going to test you on any of those structures. Alright, so we're getting towards the end of that first objective. Now we're going to talk about the top of the plasma membrane. So it's made of something called the glycocalyx. So you should know that first bullet. It's 2 to 10% of the plasma membrane's weight. It's hydrophilic because remember the heads are on the outside, and its function is for protection and cell recognition. So remember, plasma membrane allows things in and out. Glycocalyx is going to be the first thing they encounter. So glycoproteins you're going to find on the glycocalyx. So remember, proteins attached to a carbohydrate. So some examples are your amino acids, obviously because it's proteins. And then your glycolipids, which we talked about earlier, is just your lipid attached to a sugar. So galactosides, cerebrosides, remember also gangliosides. So this slide shows the membrane components again, about protein. Just pay attention to your slides because it doesn't seem to be showing the other two bullets. But the first one is your integral membrane protein. So remember, those are just integrated directly in your plasma membrane. So just think of your channels, pumps, carriers, receptors. All of those type of proteins allow things to come in and out. So the next one is your peripheral proteins. So remember, those are contributing to either the cytoskeleton or the glycocalyx. So they can be on either side of the membrane. And then you have your lipid-anchored proteins. So these are just lipids that are anchored to your peripheral proteins or your integral proteins. And those are just some examples. So here again, the picture on the right is not shown. Not sure why. Still trying to figure out the kinks. But he didn't really test on the exam. So it's just really more for illustrating what he was talking about in the previous slide. So we wouldn't worry too much about this. Maybe just read the notes. So here we have different ways that we can transport things in and out of the plasma membrane. So you should be able to tell me each type of method and give me examples of each. So anytime you're moving with the concentration gradient, you're not using any energy, so that's known as simple diffusion. Facilitated diffusion is also moving with the concentration gradient, but you're using the help of some transport proteins. You have gated channels in the plasma membrane, so these things are gated until a certain thing activates them to open up. And then we have active transport. So anytime we're talking about active transport, you're moving against the concentration gradient. So that means that you're going to be required to use energy, or ATP. So we have primary active transport, and then we have secondary active transport, and we'll talk more about those later. And then the last type of transport we have is called vesicular transport. So endocytosis, that's transporting into the cell. Exocytosis is transport out of the cell. And then protocytosis, which is a type of endocytosis that occurs by caveolae. So this picture is just illustrating the types of transport that we just saw on the previous slide. So here we have a picture of the plasma membrane. Notice on the very left we have our electrochemical gradient. Remember, anytime you're going with the concentration gradient, you're not required to use energy, and anytime you're going against the concentration gradient, you will need ATP. So if you look, simple diffusion, you see a molecule goes straight through, so it's going with the concentration gradient. If you look at the next one, you see facilitated diffusion. So again, it's moving with the concentration gradient, but you're using the aid of a transport protein. You also see right next to it the gated channels, and then you see the active transports right next to it. So that's going against the concentration gradient, and you're required to use ATP. And very right, you see the vesicular transport. So right there is an example of endocytosis. Remember, endo means transport into the cell. So now we're starting to break down each types of transport. We'll start off with simple diffusion. So again, remember that's moving with your concentration gradient, limited to small size and lipid solubility. So you should know that gases, so if he says O2, that's a type of gas, right? Then you know you can use simple diffusion. Small uncharged polar molecules. So water, ethanol, short-chain fatty acids, all those are good examples of molecules that can go use simple diffusion. And then lipophilic molecules such as steroids. So really knowing the examples, which ones can use and which ones cannot use simple diffusion. So the next one is facilitated diffusion. Remember, we're still going with the concentration gradient, but now we need the help of some sort of transport protein. So anytime you have a charged molecule, you need a carrier protein to help you cross that membrane. So ion channels are going to open in response to specific signals. You have transport proteins called uniport carrier proteins, which is shown in the very bottom right picture. So uniport means that one molecule goes in one direction straight through. Specific for substrate and inhibitors, it follows that saturation kinetics. So that's shown in the top right. And then it is regulated. And then you also have co-transport carrier proteins. So some examples of those are symporters and antiports. If you look in the picture, symporters, you're going to have two molecules entering together. And then antiports are going to carry proteins in opposite directions. So here we have an example of facilitated diffusion and how it works. The exterior is the outside of the cell, and the cytosol is inside the cell. So there you have your plasma membrane and a transport protein. In step 1, you see that glucose binds to what we call an outward-facing binding site. So it's called outward-facing binding site because it's facing the exterior. And in step 2, or right before step 2, you notice that glucose binds to that outward-facing binding site, which causes a conformational change. So if you look right after the number 2, you see that the protein, the transport protein, has changed, and now you see glucose is facing the inward-facing binding site. So in step 3, that's where glucose is going to dissociate inside the cell, and as soon as that happens, in step 4, you're going to go through a reverse conformational change to get back your outward-facing binding site. So here we have an example of a gated channel. If he asks for an example of a ligand-gated channel, the example would be the CFTR. So cystic fibrosis transmembrane conductance regulator is an example of a gated channel. You don't really have to know too in depth how it works, but in the pictures below he shows you that you have this mechanism that works through phosphorylation. So we have ABD, which is a binding domain, and then you have R, which is your regulatory domain. So in step 1, you see pKa, stands for protein kinase A, and remember kinases phosphorylate things, so this is a gated channel controlled by phosphorylation. So as soon as the pKa phosphorylates the regulatory domain, so in that second picture you see all the P's representing phosphate binding to the regulatory domain, it allows a conformational change to occur that allows ATP to bind to the ABD binding sites. So as soon as that happens, it will open that gated channel, and now your chloride ions can diffuse through. So we have two types of active transport right here. Remember, active transport requires ATP because it's going against the concentration gradient. So the very popular sodium-potassium pump, you should know which one comes in and which one comes out. So if you look at the picture below, you notice that 3 sodium come out for every 2 potassium that come in. You should know what it's inhibited by and what it's enhanced by. So certain medications are going to inhibit the sodium-potassium pump, like your beta blockers, your steroids, digitalis. So just be able to tell me all of the examples that can inhibit it, and then be able to tell me what can enhance it, like albuterol and insulin. The second type of pump that uses active transport is called the calcium ATPase pump. So calcium is responsible for muscle contraction. The purpose of the calcium ATPase pump is to maintain a low systolic calcium concentration. If we have too much calcium flowing around, then our muscles are always going to be contracted, and then we can't rest them. So you're going to find the calcium-potassium pump pretty much everywhere in the plasma membrane in most cells. And then moving on. Alright, so secondary active transport. The first example is called the sodium glucose symporter, so SGLUT1. So if you look at the picture, the very top left, glucose and sodium, they enter together, so one cannot enter without the other. That's called the symporter. If you also, when you look at this picture, just kind of pay attention to where things are. So you have your intestinal lumen, that's where your glucose and your sodium is. Then you have your epithelial cells. Notice that it's low in sodium, right? Because sodium potassium pump will pump it out. And remember, potassium goes inside the cell, so just try to make sense of it. And then in the blood, you're going to have high sodium because, again, sodium is being pumped out. And you're going to have low potassium because sodium is getting pumped in. So if you look at the very top right, it talks about digitalis. In the second bullet, it talks about the sodium-linked calcium antiporter. So the purpose of that antiporter is to maintain low systolic calcium, but it's inhibited by digitalis. So if you follow the pathway at the top, digitalis is a drug that inhibits, we learned from the last slide, it inhibits the sodium-potassium pump. So think about that. If this inhibits the sodium-potassium pump, then the sodium can't exit the cell, and therefore you have an increase in intracellular sodium. If you have an increase in intracellular sodium, that's going to lead to inhibition of that sodium-linked calcium antiporter. And if that doesn't work, then you're going to have an increase in your systolic calcium, which again leads to a stronger muscle contraction. So again, this picture is showing the same thing. There's your sodium glucose transporter, where sodium and glucose both enter the cell. And then you notice that the sodium is going to go through that sodium potassium pump. So 3 Na out and 2 potassium in. And then glucose just enters that extracellular fluid. So here's another important slide, talks about the transporter defects. This is your objective 3, knowing about your transporter defects and the diseases that are related. So cystic fibrosis, how can we get that? We can get that disease by having a defect in our chloride ATPase pump. I'm sure you guys learned about that in A&P. Other diseases, cystinuria, so impaired reabsorption of your dibasic amino acid. So if my body can't absorb my amino acid, then it's going to be excreted through my urine. So cystinuria means there's a lot of cysteine in my urine. Hartnupp's disease, impaired absorption of tryptophan, so we're not absorbing tryptophan well, can lead to diarrhea, dermatitis, and dementia. And then familial hypercholesterolemia, which is a lack of your functional receptors for LDL. So be able to tell me which disorder relates to what types of conditions. So here we talk about the different types of vesicular transport. So endocytosis, remember endo is moving things from the outside of the cell to the inside. He talks about phagocytosis and pinocytosis, which is shown in the picture below. So phagocytosis is when a cell eats something. Pinocytosis is when a cell drinks something. So you should know phagocytosis and pinocytosis is associated with endocytosis, not exocytosis. Exocytosis is just the opposite. It's moving molecules from the inside to the outside of the cell. Alright, so finally, finished with the plasma membrane. Now we're going to talk about learning objective 4, which is describing the structure and function of lysosome plus associated diseases. So first, lysosomes are the things that helps break down acid in the cell so that your cell can keep making more cells. So here we talk about lysosomes. All lysosomes are going to contain certain enzymes that help it digest certain things. There's 60 forms of those types of enzymes. Most of them are hydrolases. 2 forms are the primary lysosomes and the secondary lysosomes. So if it's a primary lysosome, you've got to kind of picture this in your head. So it says budding from the trans side of the Golgi complex. So if you picture the plasma membrane in a cell, the Golgi is located pretty much by the nucleus. If the Golgi is facing the plasma membrane, that's called the trans side. If the Golgi is facing the nucleus, that's called the cis side. So secondary lysosomes, fusion of the primary lysosome with the substrate to be degraded. And then you have some of the diseases associated with lysosomes, so you should know all three of these. So eye cell disease, and another name for that is called mucolipidosis 2, is sort of a disorder that your lysosomes are going to target the wrong thing, so it's going to break down the wrong things. Tay-Sachs, we talked about before, and then POMP. So these just talk about the lysosomal reactions. Remember, most enzymes of lysosomes are hydrolases, but you really don't need to memorize this slide. So objective 5 talks about understanding the mitochondria, structure, and function. So remember, mitochondria is your big powerhouse for energy. So it burns sugar and fat to produce energy. Alright, so the mitochondria, there's different components to it. You should be familiar with everything. So it's got an outer membrane, which is highly permeable. It's got an inner membrane, which is impermeable. So those are the key points. Outer membrane, permeable. Inner membrane, impermeable. You're also going to have the cristae found in your inner membrane, which is responsible for the electron transport chain. And then you've got the intermembrane compartment. You've got a matrix. So just be able to tell me those different parts and know the examples in each part. So here's just a picture showing you the outer membrane, matrix, and the inner membrane, also the cristae. But again, he won't have you label any type of pictures. Okay, so learning objective 6 talks about knowing the structure and function of peroxisomes plus the associated diseases. So peroxisomes also contain enzymes like lysosomes, but they degrade fatty acids and amino acids by reactions that produce H2O2, which is known as hydrogen peroxide. Alright, so peroxisomes contain enzymes that transfer hydrogen atoms to oxygen to form that H2O2, known as hydrogen peroxide. So that's how they're going to break down fatty acids, for example. It also contains catalase, which degrades hydrogen peroxide to water and oxygen when we're not using hydrogen peroxide. So peroxisomes are responsible for the synthesis and degradation of hydrogen peroxide, also for beta oxidation of very long-chain fatty acids. So you're going to learn more about beta oxidation later, but that's just breaking down fatty acids. Phospholipid exchange, bile acid synthesis, all of these are things that peroxisomes do. So below are some of the disorders that you need to know. So if a peroxisome fails to oxidize your or break down your fatty acids, then you're going to accumulate bile acid in your body. So Zellweger syndrome, that's going to decrease your peroxisome in the liver, brain, and kidney. Neonatal adrenoleukodystrophy is progressive brain damage. Infantile Refsum disease is malformation of your myelin sheath around nerve cells. So you're going to get neurologic damage. So you don't need to memorize anything here, just an example showing you where it's located. Alright, so objective 7 is describe the structure and function of the nucleus. So a nucleus, right, is the control center of the cells located in the center, regulates all cell activities, and contains DNA in chromosomes. Alright, so what are the functions of the nucleus? You can do DNA replication, replication repair, transcription, and processing of precursor RNA molecules. So you should be able to tell me those types of functions. Also, a nucleus has the nuclear envelope, which is a double membrane, and then nuclear lamina, and the nucleolus. So all of these parts are part of the nucleus. Just be able to read through these, kind of know the details about it, and you should be good. Again, just another picture illustrating what it looks like visually, but he won't have you label anything. Right, so objective 8 is explaining the structure and role of the smooth and rough ER. So endoplasmic reticulum, you have smooth and rough. That's where we package materials to be shipped. So you can think of it as your storage. The first type of ER is going to be smooth ER. So make sure you know that smooth ER has no ribosomes. The main function is to synthesize phospholipids, triacylglycerols, and sterols. Also, detoxification is a key component. Glycogen degradation, gluconeogenesis, glycolysis. So all of these are functions of the smooth ER. So here we have the rough ER, be able to differentiate it between the smooth ER. So remember, smooth ER contains no ribosomes, but the rough ER is studded with ribosomes. So this is where translation usually occurs, synthesizes proteins on those ribosomes, and then they go to the Golgi after. So objective 9 is knowing the structure and function of the Golgi complex plus the related diseases. So your Golgi is your manufacturer, right? So it receives, sorts, modifies, and dispatches proteins. Alright, so the Golgi complex has two distinct faces. So if it's facing the rough ER, then that's the cis face. If it's facing the plasma membrane, that's the trans face. So Golgi is responsible for post-translational modification and sorting of newly synthesized proteins and lipids. So after proteins are being made from the rough ER, they go to the manufacturer, the Golgi. Transport of these proteins to lysosomes, secretory vesicles, or the plasma membrane before they leave the cell. And here are two diseases that are associated with the Golgi, so just be sure to read them and know the details about them. So one, we have eye cell disease, which is also called mucolipidosis 2, and then we have hyperproinsulinemia. So here is just showing you the faces of the Golgi. So if it's by the ER, your endoplasmic reticulum, that's the cis face. And if it's facing the plasma membrane, that's the trans face. This is just showing you the Nobel Prize for Medicine. He won't test you on this. So in this slide, there's nothing really that he'll test you on, but he's just showing you that cancer cells are shown green in the Golgi. And then after treatment, the picture on the right shows them as fragmented parts. Alright, so this slide is talking about Objective 10, knowing the relationships among organelles of the endomembrane system. So really just read through numbers 1 through 6. Number 1 talks about the nuclear envelope and how it's connected to the rough ER, which is also continuous with the smooth ER. In 2, membranes and proteins produced by the ER are going to flow to the Golgi and transport vesicles. And then in 3, Golgis are going to pinch off transport vesicles and other vesicles that give rise to lysosomes and vacuoles. In 4, lysosomes are available for fusion with another vesicle for digestion. In 5, transport vesicles carry proteins to the plasma membrane so that they can leave the cell. And then in 6, plasma membrane will expand by fusion of vesicles so that proteins can be excreted. Alright, so Objective 11 talks about the structure, diameter, and function of the cytoskeleton, and also being able to discuss disorders associated with microtubules. So remember, cytoskeleton is basically your skeleton that provides the structural support for the cell and stabilizes junctions between cells. So microtubules also undergo rapid assembly and disassembly. That's all blocked or reversed by calcium, so that's a key point. Microtubules function in mitosis and meiosis. Remember, they form the mitotic spindles. Also, Chidiac-Higashi syndrome is a defect in your microtubule assembly. So, for example, delayed fusion of your phagosomes. Remember, phagosomes are what eats bacteria. So if you have delayed fusion of them, then it's going to prevent phagocytosis of bacteria. So just sort of read through all these details about microtubules and know about it, but I wouldn't focus too much on the picture. He won't make you know the different parts to it. So the thinnest type of filaments are called microfilaments. They're made of actin, so that's the key word, is they're made of actin. Each actin helix contains 13.5 molecules of globular actin. Microfilaments are for local movement and sliding movement. So they have an interaction between actin and myosin. Alright, so here we have intermediate filaments. So they are intermediate in thickness between microtubules and microfilaments. Remember that microfilaments are the thinnest, microtubules are the thickest, and so intermediate filaments are just intermediate thickness. So they primarily play a structural role. There's several types of tissue-specific proteins. So we have cytokeratins, desmin, vimentin, neurofilament, and GFA. So just be able to tell me those different types and then where you might find them. Nothing really from this slide, just showing you a picture of what it looks like. So here's a good summary of all those filaments that we just talked about. Microfilaments, intermediate filaments, and microtubules. Make sure you memorize this table pretty well. He might test on the diameter, the structure, or the examples of each. So know these well and know the previous slides talking about them. So here's a quick recap of all the cell parts. Know the nicknames, it will help you remember them, and sometimes he does give you small hints. So the last objective talks about knowing cholera, how it enters the cell, and the different ways cholera can be treated and what you can do to prevent it. Alright, so you should know that cholera is a bacterial disease that is spread through contaminated water. Cholera toxin increases because of the efflux of sodium and chloride ions. So the flowing out of sodium and chloride ions from the intestinal mucosa into the intestinal lumen is going to increase that cholera toxin. And that's going to cause severe diarrhea and dehydration, and you're going to need immediate treatment because the disease can cause death within hours. So it's present in Asia, Middle East, Latin America, India, and Sub-Saharan Africa. So luckily, cholera is easily treated. Since cholera leads to dehydration, we're going to rehydrate with sodium, potassium, and glucose, so that's option 1. Option 2 is a diet in rice. Option 3, tetracyclines. And then option 4, fluoroquinolones. So no structures, but just remember the different types of options that we have for cholera treatment. So here's your disorder table again. Make sure to read all the disorders and the conditions that follow. Most likely he'll choose cholera. So everything after this is just FYI, so no need to look at it. Good luck.